Okay, so hello everyone. It's really nice to meet you at the next TO seminar, the Tropical Ecology Online, where we will be today discussing the topic of the island biogeography. And we have got three awesome speakers. The first one is Christophe Thébaud. The second one is Amandine Vidal-Hosteng. And the third speaker will be Bárbara Freitas. And they all, so Christophe will be giving a little bit longer talk, which will be followed by the two case studies. So these seminars are organized by the Society for Tropical Ecology. And we not only do these seminars, but we organize also many other things. And for example, I will be hosting the annual international conference in České Budějovice this June. So hopefully I will see all of you in Czech Republic soon. We are also publishing the Journal of Ecotropica, which is open access and peer-reviewed of no costs. And we welcome all the contributions from tropical ecology. We are also giving the grants and awards to students, travel grants for the conference, research grants to students, and awards for the best talk and best poster at the conference. We are also having the tropical ecology student group, which is very active, and the students there are discussing various aspects of the tropical ecology and today's world. So if you are interested in what the society does, please become the members and join the student group, for example, or meet us at the conference. So once again, welcome to the 2EO talk series. And this series is happening every second Monday of the month from 3 p.m., which is now. And this is kind of a platform where the researcher, with kind of theory researcher puts down the topic, and then there is a given space to early career researchers who present the ongoing research and networks. So the first talk is typically 30 minutes, followed by two shorter talks, roughly 15 minutes, and then the joint discussion continues after all the speakers finish their talks. So today, Christophe Thébaud will be talking about ecological and evolutionary processes and which shape the diversity of the species, and he focuses on the remote islands and hyperdiverse regions, mostly in the New Guinea, where I also work, and in other oceanic and ocean islands. And then the other speaker will be Amandine Vidal-Hosteng from the same university, University of Toulouse from France, and also from the University of Groningen, who works on the island biogeography models and focuses on the theory of non-equilibrium biodiversity. And finally, the third speaker is Bárbara Freitas, again from the University of Toulouse and from the National Museum of Natural Science in Madrid, who will focus on the role of song in the speciation processes on the island birds. So this is all from me for now for the introduction, and I would like to give it a space to Christophe now first, and enjoy the talks, everyone.
Okay, thank you very much, Kaka, for inviting me to talk today in this series. It's great to be here. My name is Christophe Thébaud. I'm a professor at the University of Toulouse in southern France. I'm going to talk today about island biogeography and the origin of species, or rather my personal journey exploring some aspects of these topics in the last 25 years. So I consider myself as a naturalist with a general interest in ecology and evolution, and I have devoted most of my career to produce primary data, mainly on plants and birds, based on a combination of fieldwork and lab work. But first, I'd like to invite you to celebrate the birthdays of two scientists who have greatly shaped our current views on the topics I'm going to talk about, and much more. As many of you know, yesterday was Darwin Day, the annual celebration to commemorate the birthday of Charles Darwin on February the 12th, 1809. So Darwin is the co-discoverer of the principle of evolution through natural selection, and it is well known that islands and their species played a great role in the development of his ideas about how species came to be. A few weeks ago, on January 8th, I and a number of other people celebrated another special birthday, since it was the 200th birthday of Alfred Russel Wallace, the other co-discoverer of natural selection. So like Darwin, Wallace's contribution to our views of the world were not restricted to natural selection. Both Darwin and Wallace were great naturalists, and they were the first real biogeographers in that they used the distribution of species to crack the problem of the origin of species and the hows and whys of their existence in space and time. They both used island species to establish the process of evolution and to address questions that were key to rule out the idea that all species on Earth were the result of divine creation, such as, for example, why are there so many unique species on islands? Why are apparent affinities between distinct species so closely tied to geography? Their interest in using islands as tools to establish a theory on the origin of species is very clear from their respective travel accounts. The so-called Voyage of the Beagle by Darwin and the Malay Archipelago by Wallace, and also, of course, from The Origin of Species and the much less known Island Life, published by Wallace in 1880. As early as 1855, and this is a little known fact, Wallace published an article now known as the Sarawak's Law, in which he recognized the central role of species traits related to dispersal and geography in the processes of speciation and evolution, and he hypothesized that current and past distribution of organisms had been influenced by past geological and climatic changes. 25 later, this was extensively developed in Island Life, that can safely be considered as the first elaborate development of an island biogeography theory, into which Wallace discusses how species richness on islands is influenced by island area, island age, diversity of habitats, and isolation, 80 years before the equilibrium model of MacArthur and Wilson, which I will talk about a little later. Given the role that Wallace played directly and indirectly in the development of modern evolutionary thinking and much more, I suggest that it would be fair to have a Wallace-Darwin or Darwin-Wallace Day rather than just Darwin Day, to celebrate their joint contribution to science and society. As was the case yesterday at the Museum of Natural Sciences in Madrid. Okay, so turning now to the subject of my talk, I'd like first to give you some background about the Malagasy region, the tropical region where I conducted most of my research on the biogeography and evolution of island species before turning to New Guinea more recently. So this region is composed of Madagascar and 3 neighboring archipelagos, Comoros, Seychelles, and Mascarenes. A little bit of geological background, So Madagascar was a part of Gondwana and separated from the African landmass approximately 165 million years ago, and from India 88 million years ago, and the Seychelles separated from India between 55 and 75 million years ago when India drifted northwards. Therefore, Madagascar and Seychelles are isolated continental blocs. In contrast, both Comoros and the Mascarene archipelagos are of volcanic origin, and they derive from the activity of intraplate hotspots and are therefore geologically much younger and even quite recent. So this has important consequences from a biological perspective. Comoros and Mascarenes were never connected to mainland. They emerged from the waters as blank slates 10 million years ago, and they became available for colonization afterwards. But this was only through overwater dispersal. So while I've worked in the entire region, my research has been mainly focused on Mascarene Islands. So the Mascarene archipelago is structurally quite complex, and its geological settings is still not fully understood, with islands at different stages of volcanic building and erosion. So Réunion is the youngest island with one of the most active volcanoes in the world, and Rodrigue is likely the oldest, possibly in disappearance phase, whilst Mauritius is a highly eroded volcano, but has experienced recent major volcanic eruptions that fragmented older mountains. So there is no simple relation between age and area, for example, and the more topographically complex island, Réunion, has also been available to colonization more recently, but also this left actually less time for in situ diversification. So, I mean, the Mascarene archipelago holds a staggering number of species. So here is an example for flowering plants. So you can see 960 species in the Mascarenes. So it's a staggering number of species, extremely rich flora with many species found nowhere else, very high endemism, and for an area of about 1% of Madagascar, yet the species number is only 10 times lower than in Madagascar. So if we turn to birds and focus on regular breeders only, there is a staggering number of species too, with many species found nowhere else, but most are extinct, unfortunately. Very high endemism, which amounts to 67% and even 85% if we exclude seabirds. Here it's a rough analysis based on taxonomic knowledge of species affinities, which suggests an origin which is mainly from nearest land masses, Africa and Madagascar, but there are some odd patterns, like 1 out of 5 lineages are related to Asian rather than African taxa, but this is also a feature of Madagascar, where 1 out of 4 lineages are also related to Asian taxa. So again, based on taxonomic knowledge and recent advances in systematics, one can sort out species by colonist lineage and estimate what proportion of lineage underwent speciation after establishing on the islands. So it's probably an upper limit because we cannot rule out undetected multiple colonization in some groups. But all in all, I mean, it's about half of all colonist lineages that underwent speciation, and half of those underwent radiations that contribute a significant part of present-day diversity, highlighting the role of speciation in the build-up of island communities. So these observations raise many questions that were not unfamiliar to Darwin and Wallace about origin and diversification of island organisms. And at the time, and still today, the main conceptual framework for addressing this type of questions was and still is MacArthur and Wilson's model, published first in 1963. So in this model, the buildup of species numbers is viewed as a diversity-dependent dynamic process with a balance between immigration or colonization and extinction. And MacArthur and Wilson made two crucial assumptions concerning these processes. So first, they assume that the immigration rate for an island falls as the number of species on the island increases, and that the rate of extinction of species increases as the number of species increases. Also argued, and that's the interesting bit of it, I think, that the rates of these processes are determined by the geographical context, and that colonization, should decrease with isolation. In other words, less colonization on remote islands, and extinction should decrease with area. In other words, less extinction on large islands. In addition, they reason that the rate of species formation on islands should increase with island isolation and area. And here the reasoning was there may be less migration from mainland and more space when on large and isolated islands that will increase opportunities for divergence and speciation. So this last prediction, which explicitly relates to speciation, was not included in the core model of MacArthur and Wilson, but it was verbally described in their book. So I kept thinking about all this while getting at other things, being a population biologist by training. But when automated DNA sequencing became available in the mid-90s, I let myself convince by this man, Richard Abbott from University of St. Andrews, to switch my research interest to biogeography with the aim of producing phylogenies to answer questions about colonization history and diversification in different taxa. And that was done mainly through a very fruitful collaboration with many excellent people, including Richard. So I would like to present just 2 examples of such studies to give you a flavor of empirical phylogenetic investigations and its many rewards. So here, it's a group of plants of the genus Psiada, which is the largest genus of plants in the mascarines with 27 species. So all species are endemic. They are shrubs to small trees. They range in height from 20 centimeters to over 10 meters high for the tallest species. And the species occupy a very diverse range of habitats from the coast and up until 3,000 meters above sea level. So we put together a phylogeny after extensive sampling in the region, and quite surprisingly, we found that Psiadia species were distributed into 2 clades, which are each other's closest relatives within the archipelago and are functionally similar, yet they represent independent lineages that look similar due to convergence, here evolution towards woodiness, and therefore correspond to independent colonization events followed by radiation. So in one of the clades whose diversity is centered on Mauritius, allopatry, both between and within islands, seems key in explaining species diversity. In the other clade, endemic to Reunion, speciation seems to have occurred within the island, and colonization of a novel habitat boosted diversification with many species currently largely sympatric over the range, implying the evolution of strong reproductive barriers. My second example is about passerine birds named white-eyes, represented in Malagasy region by 10 species, most endemic to just one island, but some islands have more than 1 species. And so here, after putting together a phylogeny, we found that the diversity was best explained by 2 independent migration events, one ancient and one recent. So quite strikingly, the ancient clade branched off early in the history of white-eyes and possibly colonized directly from Asia, in contrast with the idea that islands are mainly colonized from their nearest mainland source. So in addition, within this ancient clade, phylogenetic relationship between mascarine white-eyes suggests 2 independent colonizations from some ancestral stock that possibly left no other descendants. So we investigated further one particular lineage in this clade, the so-called mascarine gray white-eyes. So it has long been known that on Reunion, the gray white-eyes present different geographic forms that are depicted here on the left. So using population genomic and phylogenetic analysis, we recently showed that contrary to what everybody thought, these different forms behave like different species, and that divergence from an ancestral stock occurred within Reunion at an unusually small spatial scale for birds. And this seems best explained by ecological opportunities and very low dispersal, which has evolved after the island was colonized. Okay, so we did this exercise for many groups and reached a number of conclusions from these empirical studies. So history of colonization can be complex, even in simple systems, and phylogenies don't say it all. Colonization may be recent and greatly postdate island formation, blurring easy to interpret relation between species formation and geological history. Colonization may also predate island formation, highlighting the importance of accounting for extinction when inferring diversification history. Colonist lineages do not always come from the nearest mainland source, in contrast to island biogeography predictions, but most do. For a given taxon, multiple immigrations through independent long-distance dispersal are always a possibility. Some lineages diversify following inter-island dispersal, others diversify within islands, supporting the idea that there is a geographical scale of speciation, that depends upon the organism dispersal biology. So time matters. Within island speciation is more important on old islands, but not always. Isolation matters. Persistence of gene flow between the source and the island can limit opportunities for diversification. And speciation matters. A small number of diversifying lineages, account for a large proportion of island species, but only when islands are reasonably large and isolated. So we could have stopped there, but we are given an incredible opportunity to draw some generalities through a wonderful collaboration with Luis Valente, depicted here, and Rampal Etienne, after Luis contacted us a few years ago for a project aimed at testing island biogeography theory predictions using molecular phylogenies at a global scale. And the idea was basically to address the classic predictions from MacArthur and Wilson's theory. So these predictions were, as I mentioned earlier, colonization rate decreases with isolation, extinction rate decreases with area, rates of species formation on islands increase with island isolation and area. So in this study, we estimated all rates at the archipelago scale using data drawn from the phylogenies themselves and including parameters like stem age, branching times, maximum age of archipelago, and using a quite sophisticated model to find the best model fitting the data. So here on the figure are depicted all archipelagos that were considered in the sample with the estimated period during which they have been available for colonization. That's a gray line on the figure, and the distribution of colonization times. So this is a blue violin with no clear relation between the two, suggesting a rather high species turnover. So figure on the right here represent the estimated relationships between island area and isolation and local island biogeography parameters. And so what we found is that colonization rates indeed decreases with isolation, extinction rate decreases with area. We also found that the rate at which island population diverge from mainland source population, what's called anagenesis here, increase with island isolation. And finally, we found that the rate of in situ species formation on islands, what's called cladogenesis here, increase with island isolation and area. So all this is very much in line with MacArthur and Wilson prediction about the important role that the geographical context can have on species buildup on islands. So based on this last analysis and insights from empirical studies, we can draw some conclusions. So first, colonization, extinction, and speciation are key processes in driving the evolution of island biotas and more generally biodiversity patterns through space and time. The role of isolation or barriers reflects the fact that below a certain threshold, movements between mainland and islands, what we call migration, also may be too high for population divergence to happen, and highlights the importance of dispersal biology that define what isolation actually is. So radiations mostly occur on large and isolated archipelagos, possibly as a result of the combination of restricted movement between mainland and islands, and or the reduction in dispersal ability after establishment on islands. For example, flightlessness in birds, and unsaturated niche space due to area and limited colonization rates. So patterns of inter-island movement within an archipelago can increase species diversity by inter-island speciation, but they can also trigger radiations if unsaturated niche space, if there is unsaturated niche space in a recipient island. So in birds, recent colonization times that imply, we found actually recent colonization times that imply high species turnover, and the quasi-absence of within-island speciation for islands smaller than Madagascar, but there is an exception, which is the mascarene, the Rhenian grey white-eye. This absence may reflect their relatively high dispersal ability. So for less dispersive organisms, understanding ecological and evolutionary processes in the context of the dynamics in space and time of the whole archipelagos, what my colleague Robin Aguilé has named the archipelago effect, and how it affects inter-island and intra-island opportunities for divergence, may be key and must be a useful avenue for more research. So that concludes my presentation. I mean, of course, all this work has relied upon the enthusiasm and hard work of many students and collaborators, in particular those listed here. And we benefited over the year from various funding sources listed here at the bottom. Okay, thank you very much.
Thank you, Christophe, very much for a wonderful talk. And we will directly continue with the talk of Amandine, and we will have the discussions and questions at the end.
Okay.
Okay, can you see my screen?
Yes, perfect, thank you.
Okay, thank you. Okay, hello everyone, thank you for having me today. So I'm Amandine Vidal-Hosteng, and I'm in my second year of PhD with Christophe Thébaud and Robin Aguilé, and my first year project was about a model for understanding how the geo-environmental dynamic of archipelagos shapes phylogenies. So as an introduction, I would like to say that the environment is heterogeneous and fragmented, and this can lead to different diversification dynamics and, by the way, shaping phylogenies. So we are interested in volcanic oceanic islands because they are much more simpler systems than continents, but also because they have a specific geological cycle. Here we have an illustration of the general dynamic theory of oceanic islands biogeography stated by Whitaker et al. And they describe this geological cycle in three phases. First, a growth phase with a volcanic activity, then an erosion phase, and finally, an submersion phase. And this typical geological cycle will induce a rare variation for the island. So when you are putting this in the context of an archipelago, you will experience the emergence and submergence of consecutive islands. So we are combining area variation and fragmentation, and that leads to connectivity variation. If I have different islands emerging and submerging, it means that the migration rates between these islands is going to vary across the archipelago's life. So a paper published last year showed that this specific archipelago structure will lead to increased species diversity compared to a single island because of this effect of this factor on the macroevolutionary processes. So if we assume that a population from the mainland colonized an archipelago, this population could be split across different islands, and it can lead to the divergence of this population and the emergence of new species. And we can show that with phylogenies. So if we assume that the archipelago is changing because of the emergence and submergence of islands, so the number of islands is variating and the area of the islands are variating as well, we can expect that depending on the moment where the species will colonize the archipelago, the species will go through different diversification scenarios. So our question is how evolutionary processes respond to archipelago-specific geo-environmental dynamics and phylogenies. And what I mean by geo-environmental dynamic is the emergence and submergence of different islands in the archipelago. To answer this question, we use the archipelago model, which is a mechanistic, stochastic, and individual-based model, which means that the macroevolutionary processes are emergent from the individual level. And it's an ecologically neutral model. That means that all of my individuals have the same probability of dying, mutating, diverge, and giving birth. The landscape dynamic of this model is described at the emergence and submergence of 4 islands. Here we have the graph of the island size depending on time, and we can say that each island in color is following the model of Whittaker and al. That means a growth phase, an erosion phase, and then a submersion phase, 4 times consecutively. And for the population dynamics, my experience starts with an empty archipelago. At some point, one individual or more can colonize this archipelago and give birth to other individuals. The depth is density dependent in this model. And so we are observing continental migration. And at the point at which the archipelago is showing more than one island, we are also observing inter-island migration, which means that the population can split across different islands and diverge and potentially emerge a new species through amergenesis. But in this model, we can also observe speciation within a population within the same island, which means that a part of this population will diverge and emerge a new species through cladogenesis. So at the end of my experiences, I can trace the entire phylogeny of all of the species that experience lifetime on the archipelago. And so our question was how the dynamic of an oceanic archipelago shape phylogeny. In this model, we have 2 factors that can bring diversity on the archipelago. First, through mainland connectivity with increasing the colonization success, which means that the species from the continent will colonize the archipelago. And on the other hand, we have the inter-island connectivity, which will split the different population across the islands and lead to speciation events. So to test each factor, we build 4 different types of archipelago. First, an archipelago which is close to the mainland, which means that we are increasing mainland connectivity. And then an archipelago which is distant from the mainland, which means we're decreasing mainland connectivity. And we also increase inter-island connectivity by creating an archipelago with close islands and decreasing it by creating an archipelago with distant islands. So it means that we have 2 groups, a group of archipelago led by mainland connectivity and a group of archipelagos led by inter-island connectivity. So our hypothesis in the point of geophylogeny is that when we are led by mainland connectivity, we increase the colonization success. That means that we expect to insert nodes at the root of the archipelago phylogeny, because when a species from the mainland comes to the archipelago, the species emerged a long time ago on the mainland. So the node is supposed to be ancient and will be close to the root of the phylogeny. On the other hand, if we are led on the archipelago by inter-island connectivity, we are increasing the speciation even by splitting the population. That means that we expect to insert nodes at the branching tips, because the new species that arrive in the archipelago will be recently emerging species. So the nodes should be close to the actual time. So our expectation about the phylogeny is that in the case of archipelago led by mainland connectivity, we should observe phylogenies with a lot of different clades. What I mean by clades is the pool of species that comes from a continent colonization, and a lot of different clades which are supposed to show less diversity, few diversity. And in the case of an archipelago led by inter-island connectivity, we should expect phylogeny with few clades, few colonization success, but a lot of strong evolutionary radiation. So I would say phylogenies that are imbalanced with one big dominant clade from the continent. To test this hypothesis, we first calculate and study the lineage accumulation through time to verify if the inter-island connectivity favors the speciation event. To do that, we use the gamma statistic, which is the statistic that helps us to see if within our phylogeny we expect more recent nodes, which means nodes close to the branching tips. In this case, the gamma statistic will be over 0. And if our phylogeny showed more ancient nodes, which means close to the root, the gamma statistic would be below 0. And we did that with the 4 different types of archipelago that you can see in color. In blue, we observe an archipelago with close islands, and in pink, an archipelago which is distant from mainland. That means that on the top, we have the group with high inter-island connectivity. The black archipelago is just intermediate distance that helps us to verify that everything is consistent. And on the bottom, we have 2 other archipelago. In green, an archipelago with distant island, and in orange, an archipelago close to mainland. That means that we have the group of archipelago led by mainland connectivity. And you can see that the more you have inter-island connectivity, which means that we're expecting more speciation events, the more the gamma stat values are high. So our hypothesis is confirmed. And on top of that, we observe that we have acceleration of lineage accumulation, 3 different accelerations that shows as bump on the curve. And these accelerations are completely aligned with the gray dashed line, which represents the island emergence. That means that in my archipelago's life, each time I have an island emergence, I'm increasing the accumulation of lineage. This corresponds to evolutionary radiation because I'm creating new space that allows the population to split and go through speciation. So it is consistent with our hypothesis, but we also tested the balance of the traits. So to do that, we use the average number of dominant clades, and what we call dominant clades is a clade that represents more than half of the diversity of an archipelago. And we can see that we also can see our 2 groups. First, at the top, the archipelagos that are led by inter-island diversity, inter-island connectivity, sorry. And at the bottom, the 2 archipelagos that are led by mainland connectivity. So the first group with inter-island connectivity shows that almost during all of the experience and for all the replicates, it shows one big dominant clade because of the evolutionary radiation. And the group at the bottom, which is led by mainland connectivity, show that the most you have islands on the archipelago, the less you will see dominant clades, because new islands brings new opportunity of colonization. That means that new clades from the continent can colonize and increase the number of clades. And by the way, increase the number of species by clade. Decreases the number of species by clade. So our 2 hypotheses are validated. And here are examples of phylogenies that I can observe during my simulation. On the left side, you have the 2 archipelagos that are led by mainland connectivity, and you can see that the phylogenies show a lot of different clades that you can see with the colors, with few species diversity in each of them. And on the other hand, on the right side, you can see the 2 archipelagos led by inter-island connectivity, and you can see that both the phylogenies are represented by one big clade that have the entire diversity of the archipelago, with 1 or 2 little clades that are persisting at this time of the archipelago's life. So as a conclusion, we can see that the first factor, the mainland connectivity, increases colonization success. And when we are combining this with the geodynamic on the archipelago, we are creating a species turnover because of the appearance of the island. And on the other hand, when we have a lot of high inter-island connectivity, we are increasing speciation events. And when we are commanding the speciation events with geodynamics, we are creating evolutionary radiation. And we can detect these 2 effects on the phylogeny. But we have to keep in mind that this study is within the context of ecological neutrality, which means that we are only observing abiotic factors such as distances, area, environmental factors. And in reality, in the real world, species have different abilities. For example, dispersal abilities, colonizing abilities, invading abilities, which means that this model could be useful in the way of understanding the relative share between abiotic and biotic factors if we use that as new model, but not as a description of what's really happening in the real world, just a simplification to understand the mechanisms that are at the origins of phylogenies. But it can be useful also in the way of using it to understand the processes behind colonization success, because it is implicit in our model that what we call connectivity is tropical pressure. And we know that nowadays it's the hot topic in island biogeography to understand the invasion of islands. And this kind of new model can be useful to understand the relative effect of environment, which is not straightforward in this kind of project. Thank you for your attention. And I would like to thank my PhD directors for this project.
Okay, thank you, Amandine. And now I would like to welcome the 3rd speaker, Bárbara.
All right, I'm going to share my screen.
We see it, but not the full screen. Okay, perfect now. Thank you.
Okay, and can you hear me well? Yeah.
Yes.
Okay. So I'm Bárbara, and usually I'm a PhD student, and usually I'm based in Madrid. You can find me at the National Museum of Natural Sciences, but now I'm in Toulouse, and I'm very much enjoying my stay there. I'm supervised by the Borja Mila and Christophe Thébaud, and now I'm going to present you ongoing work on my PhD on the evolution of song and its role in bird speciation, practical cases from island birds. So just as a simple introduction, in birds, the speciation process is usually promoted by natural and sexual selection, and these forces can drive the divergence of populations in many traits, restricting gene flow, which can lead to reproductive isolation and consequently in the formation of new species. And this animal group uses the song for many things, like individual and species recognition, but also in sexual and social communication. And this trait, the song can be affected by habitat characteristics, but also can be affected by cultural evolution. So localizations in songbirds can actually be a powerful evolutionary force, restricting gene flow and consequently promoting speciation. However, to fully understand how vocalizations may drive or maintain population differentiation, we need to account for the geographical, phylogenetical, morphological, and ecological contexts in which populations evolve. So I used to say that studying vocalizations is like making a puzzle. To see the whole picture, we must have all the pieces. And Christophe and Amandine already told you about the amazing system that islands consist. So islands are great to study because they constitute simplified and discrete and isolated pieces of the environment relative to the mainland. And when several islands are studied, they can provide the natural replicates. This is why many times we call it natural laboratories. And also bird species of island bird species often show a reduction in dispersal, which has been shown to be a key driver of divergence. So my aim is to explore the role of localizations in population divergence and consequently speciation by using two bird systems that inhabit oceanic islands. First, the common chaffinch, the Fringilla coleus in La Palma, which is in the Canary Islands, and also the Reunion grey white-eye, Zosterops bourbonicus in Reunion. But today I'm only going to focus on the common chaffinch from La Palma. So what happens in the Canaries is that the common chaffinch is in 5 of the 7 islands of the Canary Islands, but La Palma is the only island where this bird, there are 2 morphs, let's say like that, inhabiting two different that are structured according to the habitat. So we have the cloud forest here represented in blue, and we have the pine forest in orange. And chaffinches from cloud forest are usually bigger and also have more orange in the breast than the chaffinches from pine forest. And Maria Recuerda from our group used the genome environment association analysis and showed that there is a adaptive genetic divergence between the habitats, specifically related to the NVI, which is a measure of greenness. So actually there is evidence for adaptive divergence to the related with the habitat, and my aim was to assess the importance of the role of song to drive or and then maintain these diversions. So I went to the field I recorded birds in multiple locations in these 2 types of habitats, and then I used bioacoustic software to see the spectrum, which is the image of song that you can see here. And well, the song actually looks like this. Sounds like. Well, I don't know if you could listen. I hope so. So and then I measured some acoustic variables mainly related with time and frequency, like maximum and minimum frequency, bandwidth, duration of the song, the peak frequency, and also the sea level rate. And then we found out that this from all the variables that we measured, chaffinches seem only to significantly be different in the lower frequency. So with chaffinches from cloud forest singing at lower minimum frequencies than chaffinches from pine forest. And then we used tree cover to actually check if this was related with habitat and not a biased categorization made by us. And we actually see a negative trend. So here in this plot, each point is one individual for which the multiple songs were average. And here you can see that for higher values of tree cover, the chaffinches usually sing at lower minimum frequencies, and those are the chaffinches from cloud forest here set at humid. And this is actually consistent with the acoustic adaptation hypothesis that predicts that in dense habitats, low frequency sounds thrive more efficiently than in open habitats. This is due to mainly things like reverberation or absorption of sounds by the vegetation. But this hypothesis also predicts that other variables should be different in specific ways, like the birds in or species in dense inhabiting dense vegetation habitats should also sing at lower maximum frequency, have narrower frequency ranges, have longer songs, and a slower pace. And this is not happening with chaffinches songs. And actually, when we look posit in the literature, there are many studies supporting this hypothesis, but that there are many others that not supporting it, or with mixed results, like in our case. So this means like having one or two variables supported by the hypothesis, but others no. So we now are analyzing and studying this hypothesis in more detail through a meta-analysis. But morphology and body size could also be a factor affecting this relationship because heavier species are supposed to sing at lower frequencies, and heavier species in our case are chaffinches from that inhabit in cloud forest. So morphology could be acting as a confounding effect in this whole thing. So in the future and with large sample sizes, we aim to confirm this pattern of birds from cloud forest having lower minimum frequencies, but also correcting for the morphology. And besides this, I also assessed if there was behavioral divergences between those populations by using playback experiments, and this was more or less the playback experiment setting. And just to explain you quickly, so I put a speaker attached to a pole close to a tree or a branch where chaffinches could use as perch. And then I was apart taking notes, so I played a song for 1 minute, and then I resisted any behavior for 10 minutes. And each bird was only tested once, and the song that I played could be one of 4 treatments. It could be a song from an individual from the same type of habitat, from a different type of habitat, from chaffinch from Iberian Peninsula, so it would be a distant related chaffinch, and could be white noise that was used as control. And here in this plot, you can see the 4 variables that we measured. And all these variables are supposed to be related with aggressiveness to intruders, and so closest approach distance, number of times calling, time to first approach, time spent within 4 meters of the speaker, which will be like this radius. And what we found was that chaffinches replied differently, so almost didn't reply to songs from the Iberian Peninsula, and didn't react at all to the control, which was expected. And this is represented by gray and black. But there was no differences, no significant differences between reactions of the same habitat in different habitats, represented by these two colors. So, which suggests that birds from both populations still recognize each other as potential intruders closely related. In the future, we want to expand this the area where we did the playback experiments, and also correct for distance to actually assess if there is character displacement, which would be individuals being more aggressive to songs from close birds than from farther away. And so I want to thank my supervisors, Rosa and Christophe, all the people that helped me, also the funding agencies, and thank you for watching the presentations. I'm happy to reply to your questions now or after it.
It's perfect. Thank you very much, Bárbara. It was a really nice talk again. And I think all three of them very nice accompanied each other. And first, I would like to ask if there are some questions in the audience to start with. Maybe you can just speak up or raise your hand. I don't see anything, so I will ask by myself first. So maybe my first question will go to Bárbara, as I see her here. So I was wondering, is it always, or is it typically first the sound or call which makes the species to distinguish each other if they are like this separating in the different habitats? Or are there some bird species which try to differentiate in the habitats because of the body size or colors? What goes first to diverse?
Well, that is a difficult question, almost like as the which comes first, the chicken or the egg. But it could be, yeah, some species like is a consequence of the difference in morphology, and was like more or less what I tried to explain. Like if they are bigger, they also tend to have differences in songs just because of the differences in mass or size of the body. Or then the shape of the bill can also be related to that. So it can be a consequence of adaptation to the habitat or to diet, actually. But also can be like can be the first thing to happen. For example, when there is cultural evolution, for example, or memes like different populations having different types or different syllables, and then within the same populations they prefer those types of songs or syllables, let's say, and then that this although they are not different, and that acts like a barrier, and they don't reproduce anymore. So that can start actually the whole process of the speciation as well. So, but it's a bit tricky to know exactly.
Okay, thank you very much. Well, there is one question now, but I don't know who is that.
It's me.
Oh, okay.
Yeah, I had a question for Christophe. Thank you for the nice introductory talk. I was quite surprised that the endemism in Madagascar is lower compared to the islands around. I didn't know that before. And you explained that the species come also from further away to the islands around Madagascar. And is this the main reason why we see higher endemism on like the Macarene Islands compared to Madagascar, which is much older? And yeah, or did I understand it right?
Yeah, I mean, in a sense, I mean, the level of endemisms are not that different, between Madagascar and surrounding archipelagos. Actually, The level of endemisms are also quite high in the Comoros, although slightly lower than in the Mascarines. And they are actually much lower in the Seychelles, probably because of their small area and because there's been quite high turnover, extinctions, and then recent recognition. Yeah, I don't know if I would argue too much for possible causes for that may explain differences between the neighboring archipelagos. I think it's just that they're all function similarly. I mean, and in many groups, I mean, also in the Mascarines, there's been substantial species radiations. So of course, when you have radiations, this adds up, to the number of endemic species. It makes more endemic species. There's been also radiations in Madagascar, but although we don't have a precise figure, if you take birds, for example, I mean, there are many birds in Madagascar are shared with neighboring Africa, in fact, and some groups, for example, like the Zoostrops, have not diversified within Madagascar, but they have extensively diversified around Madagascar on the archipelagos. Similar situation in birds also is found in foodies, which had only 2 species in Madagascar, but multiple species on the islands around. So yeah, I mean, it's I think perhaps the most striking fact from the Mascarines is the number of species. It's just 1/10. It's 1/10 of what's found in Madagascar, even though it's 100 times smaller. So which means that there is a concentration of species, if you want. There is a concentration of endemic species per square kilometer in the Mascarines. That's definitely way outside the range, even of places like Madagascar.
And when you talk about endemism, you mean subspecies endemism, like which level?
What in the figures I presented, this is species level endemism. Yeah.
Yeah, that's incredible. Like how still a bit higher it can be than Madagascar.
It's probably it's probably what do you say, an underestimate, given that there are some groups, including in plants, where the systematics has not been worked out properly, and so right now the species that are found in the Mascarines are considered as conspecific to species found in Madagascar, but unless there are some data on their evolutionary relationships, is a bit cryptic diversity, in other words, could also kick in and modify the statistics. But anyway, it wouldn't change much, the take-home message, which is like high species number and really impressive level of endemism at the species level.
Yeah, thank you.
Is there some more questions from the audience? Okay.
Yeah.
Melanie?
Oh, sorry.
Go ahead, Claudia.
Thanks. Sorry. I wanted to ask a question to Bárbara, and I was wondering if you have observed or heard the same differences in islands that had similar habitat types differentiation, such as, for example, Tenerife?
So in Huapalma, in the Canary Islands, Huapalma is the only island where actually there are these 2 morphs. So because this is the only island where there are the 2 types of habitat well preserved. So in the other islands, there is no pine morph, let's say. So it's only the cloud forest. So we don't actually study this within island diversification. What is studied is more the difference between island mainland and then between all the islands themselves. Yeah.
Thanks.
So.
Okay, I think now Melanie.
Yeah, thanks for these really great presentations. I certainly learned a lot. My question is more towards this this fact that quite a bit of the fauna of this area has been gone extinct, and that you have used quite sophisticated modeling, both statistically and also with this individual-based model. Do you have any grasp on, or yeah, the question would be like, is there more extinction that we don't see in the empirical data that you might actually predict from your modeling work had happened? Because obviously we don't pick up every extinct species, of course, but we might predict that there have been more.
Yeah, although I mean we put some effort to include actually I mean recently extinct species or species that have gone extinct due to human causes. We put some effort to include them in the analysis. So in a way, I mean these are extinction rates, you know, discounting for the effect that humans had on recent extinction rates. So it was more like to, you know, see what, I mean, how the extinction rates over the long term may have driven these patterns. But for sure, I mean, it's not accounting for recent extinct species would have created a strong bias, you know, in our estimates, which is why we made this effort to include them. So we had some kind of little tricks, you know, to include them, even though we didn't have molecular data for these species. But knowing the relationship with some extant species, we could include them in the analysis.
Okay, thank you.
Okay, I would like to have actually a question to Amandine, because I was wondering also about the models and how specific they are to the island system as such, and how they could change, or whether they would be exactly applicable or different for those so-called sky islands, where you have the habitats on the mountains which are separated by some habitats in between. Would it work the same way, or would there be some difference?
It could be different. Actually, for the moment, it's just applicable on the island system, because like we are following, you know, the Hampshire curve that describes the island variation. But actually, the function of the area variation could be whatever you want. You can like implement it as a new function of, for example, the glaciation level that will increase within the mountains. So the area variation will be different, but actually the model works exactly the same. You just have to change the carrying capacity function, so you can apply it to every system you want, as long as you know how the area is variating and the fragmentation is working.
Okay. Okay, thank you. Do we have some more questions? Oh, in the chat, I see only the messages that the talks are interesting and or very nice, but no questions. Okay, so in that case, I believe that the speakers exhausted the audience. Thank you very much once again. And before we would, oh, okay, some clapping. And I would like to share once again the slides. I can hear, yes. So just to, because the talks were recorded, so here under the conferences on the web pages of the society, you will be able to find the recordings and also the older seminars to share with your colleagues. And in a month, we should have the next seminar, which will be focused on the metabarcoding. It will be 13th of March. And then once again, I would like to invite you for the conference of the Tropical Ecological Society, which will be happening in České Budějovice in Czech Republic, and later this week we will be opening the registration and the abstract submission, because we have got already selected the symposia for the conference. So thank you very much once again for attending and giving the talk at the Tropical Ecology Online seminars, and see you in a month.
Thank you very much, Katka. Thanks.
Thank you, Christophe.
Thank you. Bye-bye.
Thank you,
Bye.
everyone.